Hello everyone, today I will be talking about my project, which is the expression of the isPETase gene in E. coli for the degradation of polyethylene terephthalate or PET. So this gene encodes for the enzyme PETase that catalyzes the degradation of PET into its monomers. By purifying and isolating this protein, we will be able to metabolize plastic waste very efficiently. So for my project, the plasmid vector that I'll be using is PET21B, which is used for high expression of a protein in E. coli. These are some of the features that it has that make it suitable for my project. The T7 promoter, for example, allows for strong inducible high level protein expression in E. coli. The lac operator allows for the inducible expression of my protein by the addition of IPTG. The ribosome binding site allows for a more efficient initiation of translation, and the multiple cloning site has multiple restriction enzyme sites that allows to insert our gene of interest, the isPETase. The his tag will be added to my protein so that affinity chromatography can be used to purify the protein, and it also has an ampicillin resistance marker for selection of our colonies. So the cloning strategy that we will be using is as follows. This is the first step, vector preparation. So we will take our cDNA and we will amplify it using PCR. We will be adding the restriction enzyme sites to the DNA using the primers that we have. The restriction enzyme sites are two different sites, BAMH1 and XHO1, which have non-compatible sticky ends that ensures the directional insertion that happens within the plasmid is in the direction that we want it to be. So once we have our PCR product, we will run it on a gel just to make sure that it is the correct DNA that we want to be inserting into our plasmid, which is around 890 base pairs. We will then digest it along with the vector separately with our restriction enzymes, and then later ligate them with T4 DNA ligase. Over here we can see the DNA having been inserted into our plasmid. So then the second part of the cloning strategy is bacterial transformation. This is just a standard protocol for how we do it. We will be using one-shot top 10 chemically competent E. coli cells, which have been prepared for the uptake of our plasmids. We will be using heat shock, and we will then grow these bacterial colonies on a plate with ampicillin and then transfer them to a liquid culture for the next parts of our experiment. However, it's also important to do quality control for our experiment, and for that we will extract the plasmids using a mini prep protocol, digest it to separate the DNA insert as well as from the plasmid, and run it on a gel. If this is the correct plasmid, we should see two bands, one with our insert and one with the vector, and later we can also send it for Sanger sequencing to ensure that this is the correct protein or the correct gene sequence that we have extracted. Here I have shown how I've designed a primer that starts from the T7 promoter, and using this, the Sanger sequencing method can go forth and give us the sequence of the DNA. Finally, we will be doing protein expression and purification. Now, with the plasmids that we have grown in the E. coli colonies, will be purified and transformed into the BL1DE3 competent cells. These are specifically used for the expression of proteins. The protocol will be very similar to before. So this is how they'll be transformed and grown. And now IPTG will be added to them to induce the expression, and once we have protein being produced by these cells, we will purify that protein using affinity chromatography using nickel beads. Once we have our protein purified, we will verify it using STS page just to make sure this is the correct protein, and thus we will have our product. These are my references, and thank you for listening to me.